to one and all. Welcome students to the Department of School of Life Sciences, GNU main campus. I am hereby very glad to welcome you all in this department. On the behalf of the School of Life Sciences, I am presenting my introductory lecture for the microbiology. So let's deal with the introduction to the microbiology. This course actually deals with the study of microorganisms. These microorganisms are present in nature in all, but we cannot observe it with our naked eye. Therefore, they are called as microorganisms. This is the study of microorganisms such as bacteria, viruses, archaea, fungi, and protozoa. These are actually having the two aspects. One aspect is very beneficial to us and the other aspect is pathogenic. So we will be dealing with that aspect afterwards. First, we want to have a note what is an important scope of this microorganisms. First, we are dealing with bacteria. Bacteria, what are bacteria? They are microscopic single-cell organisms that exist in their millions in every environment, both inside and outside of other organisms. For example, on our skin, we have a lot of microbes present, which are both beneficial or sometimes injurious also. Because the basic nature of this bacteria is that they will first establish itself as a single cell, and that is not at all observed by the naked eye. For that, what we have to do? We have to grow it in our labs and then analyze it and then see either it is beneficial or it is pathogenic. Right? So these are the microbes which you are seeing on the screen. These are single-celled organisms which are present in millions, what I have already told you. And next is fungus. Fungi, which is a plural word, is a member of group of eukaryotic organisms that include microorganisms like yeast and mold. This yeast and mold has a very long impact on the economic value. These are responsible for production of alcohol in the alcohol industry from the common substrate of any type of sugar compounds which you are taking. This is actually a very general statement which I am giving you. The substrates are specific for the specific production, right? Similarly, for molds, these are also sometimes beneficial, sometimes not. Molds, for example, Aspergillus niger, they are critically involved in the production of citric acid. Means they are all the microbes are having both the aspect of positivity as well as negativity. Here you can see this yeast, which is as a single organisms, they are also having the colony on the same, and sometimes they are having different other forms too. For that, what we can see is that here you are seeing this is called as actually yeast. Next part, which is a multicellular part, we can see the mushrooms. Mushrooms are obviously it will have two forms. One is a edible form, another one is non-edible part. Non-edible part is very toxic in nature. If taken in minor concentration, can go for the health injury like anything and can cause death even. Second aspect, this mushroom, which is edible mushroom, is having a very good content of protein, and they are very helpful for us. Even they are anti-carcinogenic. They are having typical compounds which is going for their carcinogenicity neutralization. Next, when we are coming to the virus. Virus, what is virus? We know they are small particulate matter actually, which is not toxic by itself. For that, what we have to go? They have to go with, they require some host for their pathogenicity. And a virus is a submicroscopic infectious agent that replicates only inside the living cell. They required the host metabolism for their growth and reproduction. right? Now going with that, bacteriophage, which you are seeing here, they are having various parts in it. These parts are tail fibers, pin, sheath, nucleic acid and capsid. This capsid is an outer cover and the nucleic acid. Both in the combination, they will go with different aspects of pathogenicity. Nowadays we are seeing the coronavirus like anything. This is going with the hamperment of the whole world, and because of this, their nature on the surface pathogenicity, they have a shift antigenic and shift antibodies. They are not produced according to that. For that, we are having different other forms. Therefore, we are not able to provide with a vaccination of the same. Right, we are seeing here the human viruses like herpes virus, HIV virus, Ebola virus, adenovirus, influenza virus and hepatitis B. Now coming to protozoa, they are informal term which is a group of single cell eukaryotes. They are either free living or parasitic. Free living, obviously they are non-pathogenic one. They are going for the proper maintenance in the ecological balance. One important thing is that all these microorganisms, if they are not present in the environment, then our living cells cannot survive, because they are mainly involved for the decomposition purposes as well as for the different type of treatment plants. For example, waste treatment plant. These microbes, they play a very important role in the waste treatment plants. Waste water treatment plants, they are dependent on these microbes for their removal of BOD, which is a part of a secondary treatment plant. We will discuss it further. So, we are coming back to the protozoans. Protozoans, this is an informal term of a group of single cell eukaryotes. They are either free living or parasitic, which feed on the organic matter such as other microorganisms or organic tissues and debris. Here you can see the different examples of the protozoans. These examples are from top left, we are seeing it, that is Viphrasma japonicum, and going with different type of Giardia murius and the flagellate ones. Here you are able to see the different forms which is going with dealing with different type of protozoans. This is just the example. There are a lot of more protozoans which have the ecological importance too. Right? Now coming to the very important part of the history of microbiology. History of microbiology. Here you are seeing a person with this curly hairs. He is called as father of microbiology. He is Antony von Leeuwenhoek. In his lifetime, he has very much contribution. Actually, he was a Dutch merchant, and in his lifetime, he has produced about 250 to 300 microscopes, which are actually not the microscopes. They used to make a different combination of grinding of lenses, and from these lenses, he used to observe different samples. For example, saliva, pond water, honeycomb, any blah 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 blah. And by this, he used to see small organisms which are present in this turbid water or turbid solution, and they used to move from one place to another sometimes, right? So what he called it as animalcules, which is animal-like molecules. Next is Louis Pasteur. Louis Pasteur, he is called as father of modern microbiology. Why he is called as father of microbiology, we will discuss. Next is Robert Koch. He is called as father of bacteriology. These are some of the milestones which you should remember before studying the microbiology as a subject, right? Just now I have told you about the Leeuwenhoek contribution. In 1676, Leeuwenhoek discovers animalcules. In 1798, Jenner introduces cowpox vaccination for smallpox. What he has done? He has taken the from the cowpox the mate which is not able, they don't have the smallpox at that time, where the rest of the world is having the smallpox. Smallpox is very dangerous disease, right? So what we have to do, we have to have some preventive measures regarding that. What he thought was that he observed the fact that the cow mates, those who are going for the milk generations from this cow, they have some pustules, they have inflammations in between their fingers. That pustules is filled with the lesions. What he thought, if these lesions has to be introduced to a person to a smallpox, then the person is recovering from that of smallpox, right? So for that person, he has done this part. Next come with the Schwann and Sheldon cell theory. You well known it. Pasteur shows that the lactic acid fermentation is due to the microorganisms. In 1961, Pasteur has a lot of contributions in the field of microbiology. Pasteur shows that microorganisms do not arise by spontaneous generation. Similarly, Joseph Lister, due to his published work for the antiseptic surgery, today we are having a disinfection proper in the hospitals so that surgery can be carried out. For that, he used phenol as an antiseptic agent, right? Next we come to Robert Koch. Koch is well known with his Koch postulate. Koch postulate comes with the anthrax produced by the Bacillus anthracis. Koch has given the four postulate. What he was studied in his lifetime, very important aspect, which is called a germ theory of the disease, is that a person or a cell or a living organism, when infected by any of the forms of disease, should, when it is isolated, that pathogen when isolated in the pure form, will come in the isolated form in the plates, right? From this plate, if we are going, after purifying, incorporation of the same pathogen into a healthy individual, it should produce the same symptoms of the disease as that of the old one. This is famously called as Koch's postulate, or you can say it is the germ theory of disease, right? Next, we are coming with Petri dish. Petri dish on which we poured the media. What is media? Media is that component, because in nature there are a lot of microbes present. We want to cultivate it in the lab, then we have to cultivate it in the media. This is given by Petri dish, was given by Richard Petri. Next. Now, we have seen about the Leeuwenhoek, right? For in his lifetime, he has produced different type of combination of lenses. After that, there was a hold between the starting of the new era of microbiology, right? At that time, scientists was debating about different forms of life. Life, they have been originated from a living cell, or the life has been originated from a pre-existing cells, or abiogenesis, that is, life may not have been existed from the living, but may have been generated from the non-living forms, right? These questions has to be answered. Some scientists, they were in favor of the abiogenesis theory, that is spontaneous generation theory. What is spontaneous generation theory? Spontaneous generation theory deals with life may have been originated from the abiotic environment and non-living forms. They are responsible for the generation of life. This is spontaneous. And what is biogenesis? The scientists told, the group of scientists, they told that life has been originated from living cells. These are the two parts which exist for the debating between the scientists, right? So, the questions arises like, spontaneous generation of the microbial life possible or not? What causes fermentation? What is the cause of the disease which we now and then encounter? Previously, there was not proper interpretation, so the pathogenic effect of various diseases are a lot, right? Now, what I have told just now, that is spontaneous generation, Life, biogenesis we are dealing with, life arises from the pre-existing life. And abiogenesis deals from non-life forms. Needham and Splanzini, Pasteur, these are some of the scientists which were in favor of that there is no spontaneous generation theory exists actually. This was a debate between the scientists, and different experimental proofs were established to prove that life has been originated from a living cell. Classifying the microbes is one of the important criteria. Until unless you classify the microbes, we cannot go for their proper identification. This was given by Carolus Linnaeus, who developed a system of general classification, and this was the initial part in the field of taxonomy. Microbial cells, which we have seen till now, is basically of two types. Either they are single celled, which is called as prokaryotic one, or they are eukaryotic. There are 6 main classes of microbes, which we will discuss one by one, but this is a generalized forms. They are fungi, protozoa, algae, prokaryotes, bacteria and archaea. Archaea are actually primitive bacteria. It has been proved that this previous bacteria, they are responsible for the generation of the new cells of eukaryotic system, which we called as the generation. This generation has been established due to this archaea, from archaea, and with different other forms of the living cell, multicellular organism may have exist. Actually, archaea, they are found in primitive conditions, that is either extreme conditions of the environment, either very high heat, very cold water, very high pressure. According to that, they are named. Right. Next comes the helminthes, which is a parasitic worm, and the viruses, which are not cellular, as already explained to you. After that, there was the explosion of interest in microbiology. They are going with this questions that the spontaneous generation theory, which we have already dealt with. Here we are seeing one experiment given by Francisco Reddy. You can see here what happened is that here you are seeing the maggots or flies, which is having, this is a meat, we can say pieces of meat. This pieces of meat, they are having different type of flies over here. Right, they growed, and when we covered it, the flies were not present at that part. Right, after that, what happened? Some of the flies, after covering it, have covered the surface. This surface, when we are going with the open jars, you can see here flies. This is open jars. There is a lot of flies present over here, which is going for the reduction of the substratum. Here the substratum is meat. After that, what happens is that this flies, again, we are having the 3 parallel set of experiments over here, right? So here we are opened the flask, here we have closed it. And tightly sealed flask, the flies are outside and there is no reduction of the substratum here. Right, and here we are having a cover with a net, and this is a tightly bound jar. Here we are having the net, and this net will have the different forms of flies which is gathered on the surface, and they lay eggs. And these eggs are responsible for the generation of new flies, not this substrate which is going for generation. So this proves that he was not very much able to explain that at that time. This was further given by Louis Pasteur, who finally concluded with the generation of biogenesis. Right. So what happened is that here you see the flies. These flies, they will go with the production of eggs, and from this eggs the new generation will rise. Right. So this is one of the experiment which explain that life has been originated from the pre-existing life, not from the abiogenesis. Right. Now, why the fermentation is there fermentation of any use of the substrate of the sugar, and there will be production of alcohol. Pasture has contributed a lot because most of the wine production in the different type of industries were suffering at that time. Pasture has observed that this wine, it is spoiled because of the production of lot of acid, and it is not fit for the human consumption at that time. So what he has done, he has given a concept, a very important concept of pasteurization. That is suddenly heating something and then cooling it. This is now used in various industries of juices, milk, specifically for the milk, and for a long time we can have the milk ready for the use, right? Now you are seeing the Lewis Pasteur, which I have told you, with different type of forms, and here we are seeing Jenner, and this is Jenner is vaccine giving a pustules lesion indulged into this young boy of 8 years, and he has been recovered from that of smallpox, right? This is a form. Different contributions which are going with different type of disease we have already dealt with. They have given a revolution, and there is a lot of industries come into the play, and enzyme production for biochemistry field, different type of fields come into the play, and they show different scientists from the corners of the world. They have participated for the production and the disease controlling agents, right? So here we are seeing some of the important contribution, that is Samuel Willis. He has given hand washing to reduce the infection. Lister, he developed, I have already told you, the phenol solution for the surgery, and this solution is used to sterilize the surgical instruments, and significantly they reduce the surgical infection. Eldridge, magic bullet led to the field of chemotherapy. Jenner smallpox dealt, and this Nightingale's antiseptic techniques and nursing practices relate to the field of nursing. This now studies that there has been a different type of occurrence, distribution, and the spread of diseases in the population. Means how the disease they infect. Sometimes they are contagious, sometimes they are non-contagious. This type of concept was first given by Sloan. Now we are coming with the modern age of microbiology, examining the basic reaction. What is a modern age of microbiology? Modern age is nothing. From the old experiences, we are trying to establishing the facts. Obviously, for any experiment to set up, we should have a hypothesis, theory, and the experimentation. These are the three important key parts for the application of any type of scientific facts. The scientific facts require a thinking, a processing, a thought, what to be done, what thing we have to analyze, and where we have to execute it, right? For that, what we are happening is that we will first think it, we will write it, we will thought process it, and then we have to go for what are the nicks and prawns for the particular things to be dealt with. And then after establishing the facts in the lab, then we can execute it for the major or a bigger experimentation part, right? I think by that you have been a knowledge about a basic concept, a basic knowledge, you can say, a basic knowledge for different type of the things which is happening in the world of microbes. By this I am concluding my lecture today. Next time we will continuing with the same matter next time. Thank you so much.